My talk begins with a kind of a personal account of my beekeeping experience. I've been keeping bees for 10 years. My first 2 years were a disaster, like they can be for many people. And so for that reason, I engaged in some education with FIBCA, the Federation of Irish Beekeepers Associations. And very shortly after that, I became interested in genetics. So I joined the Native Irish Honeybee Society in 2012. And a few years after that, Micheál McGillicuddy asked me to write a pamphlet about genetics, which has recently been incorporated into the book that NIHBS published. So I'll introduce you to my area and my bees and my beekeeping, and then I'll outline some basic genetic precepts, very much to do with practical beekeeping, which I hope will be of benefit to anybody starting on their journey of selective breeding and improvement. So the aims of this lecture then is just, as I said, give you some background information on where I live and what kind of bees I keep, and the desirable traits that I need, which will match the environment. Then I'll pose the question, why study honeybee genetics in the first place? And then we'll take a look at that famous molecule DNA. We'll then take a practical view on the mating behavior of Apis mellifera and talk about how that impacts on how genetics impacts on that. And then I'll bring you through some basic terminology that's genetically related, but it's more relevant to bee breeding. I'll talk about genetic variation and how the honeybee is designed to introduce lots and lots of variation through reproduction and swarming and colony reproduction. I'm going to talk about a little bit about sex determination and inheritance of dominant and recessive genes. So that it's really a very rapid kind of bird's eye view of genetics, and it's only going to be genetics that's relevant to bee breeding and bee improvement. And then I'll recap with some take-home messages, and I'll give you an update from the Native Irish Honeybee Society, which was sent to me courtesy of John Greenaway. So I live in East Mayo. It's one of the wettest counties in Ireland. To the south is County Galway, where the Connemara Mountains regularly would receive about 2,400 millimeters of rain per year. Meanwhile, on the east coast in Dublin, we get about 1/3 of that rainfall, 800 millimeters per annum. So there's a big difference in climate, even comparing the west and the east of the country. And that's why I always say to people, you know, your area dictates the kind of bee management that you practice. So somebody in Dublin can't really, we may share the same species of bee, but our management styles have to be very different. Our temperatures in the spring and autumn hover at around 10 degrees Celsius, and our summer averages, apart from last year, which was an exceptional year, are between 14 and 16 degrees Celsius. So the warmest month is August, and we might hit temperatures of 18 degrees. There's no week really in the year where we wouldn't normally expect rain. Again, last year was an exception. And January and February are the coldest months, 47 degrees. I have hybridisation to the north and to the south. There are known beekeepers importing buckfast bees, and we have a big problem on the frontier between Sligo and County Mayo, so much so that a lot of beekeepers who were in one particular club to the north of me who wanted to keep the dark bee came to our club. And so I have a lot of first-hand information about and anecdotal stories about how tough it is to maintain the black bee about 20 to 30 minutes drive north of me. Again, 20 to 30 minutes south of me, we have another cohort of beekeepers who are keeping buckfast bees. But south of that, again, we have Connemara, which is one of the great strongholds of the dark bee in the country. And there are many now, thankfully, as people begin to join conservation areas. For me personally, I can expect to do my first inspections around the 10th of April. When I open up on the 10th of April, I'm likely to see the first drone brood. And for me, that signals that 40 days hence, swarm season will begin. So the thing is, the message I have here is get to know your locality, document the pattern every year, and that will give you a lot of confidence then in when to initiate queen rearing and when to expect swarm control measures will have to be done. This is just a snapshot of my area. So as you can see, it's very rural. The landscape is very rugged. I'm not sure what altitude we're at. We're quite high up. And even 2 miles away, I have an apiary which is closer to sea level. And that apiary, developmentally wise, would be 2 weeks ahead. So again, know your apiaries and your bees and the pattern. On the other hand, even though it looks very uninviting on a wet and a windy day, by midsummer we have a huge range of wild flora, which supplies a very diverse and early range of pollen, and that continues right the way through the season. So I rarely have to use any supplementary pollen or fondant. In fact, I deliberately try and have bees that don't need artificial feeding. So this would be a typical frame of pollen in the summer. You can see the khaki green pollen is from the blackberry, but we have a whole diverse range of pollen there. And I'd expect to see at least 2 frames like this on a weekly basis. And when you have plentiful pollen like this, no crops, no pesticides, no neonics to worry about, you can expect lovely frames of healthy brood by early May. And as I said, I always count 40 days from when I see my first drone brood, and then I can nearly predict to the week when swarming begins. And that pattern is constant every year. As you can see, this is not wall-to-wall brood. I don't really want to see wall-to-wall brood. If I do, then I know I've got hybridization. The queen is here on the top right-hand corner. She would be an example of a queen that I started with maybe 6 or 7 years ago. I wasn't really in control of what bees I could source. They were hard to source, in fact. So, and then I had a friend who brought bees in from the north, and his queens were mahogany. They were native queens. They produced dark black bees. And queens like this, I noticed after about 2 seasons, were likely to produce 50% jet black queens, as I called them, and 50% queens of what I called mahogany in color. I had no banding, no bandiness, no buckfast. So at this point, 7 or 8 years ago, this was a turning point for me, noticing these differences. And this is when I got interested in genetics. So to maintain colonies of bees in this area, I have certain desirable traits. Again, when I started reading about genetics and desirable traits and drone breeding, I got a bit bogged down, and I just wondered, well, what's the difference between the desirable traits that you need for breeding drones and breeding queens? And in the end, I just demystified it, and I said to myself, whatever makes a good drone producing colony is bound to make a good queen producing colony as well. So the traits I look for would be docility and steadiness on the comb. Obviously, the temperature has a bearing on the temperature of inspections has a bearing on how bees behave. They don't want to be interrupted in cold weather, but you saw what our average temperatures are like. So it's really important that I have docile bees. Also, winter hardiness is essential. We have like a 6-month winter here. And in recent years, you could say that we're just cleaning up colonies that have died out. No sooner have we tidied up those colonies than April hits, and the month of April could be spent tidying and getting ready for the season, and the season hits then in May. So it's a very quick turnaround. The effects of the winter seems to stretch right into April these days. I don't want bees that are prone to swarming. I do use swarm cells for propagating queens, but they're I noticed that the dark bee doesn't produce a prolific amount of swarm cells. So I don't feel as if I'm breeding swarminess into my population. but I do like the queens that come out of swarm cells. I used to graft, and I tinkered around with lots of different ways of breeding queens when I was selling nooks to beginners and stuff like that. But I found it was tricky enough to master grafting. I didn't have the time to devote to it, and the queens, inevitably, a lot of them got superseded. And that's a problem I hear about often. And I often wonder, is it the way we're breeding queens en masse in huge numbers? Do we need to look at that? I've given up. I don't sell bees to beginners anymore. If I had to, then I probably would have to master grafting or some other more natural method of rearing queens. But for me, where I am now with my beekeeping, I use swarm cells. So in other words, I don't want high maintenance bees. I want robust, strong colonies going into the winter. So again, when I have my swarms control being done in May, I can guarantee that I have queens mated and laying by early June. And in fact, the 31st of May is a goal I have every year. I like to have 1 or 2 queens mated and laying by the 31st of May. And that means that if I start a new colony with those queens, that those colonies will have filled a national brood box. National hives are what I use, timber and polystyrene. So that new queen will have filled the box, and the colony may even have started filling a drawn super, or be drawing out a drawn super by the end of the season. Good spring buildup on low levels of varroa, chalk brood, and varomorpha, which was formerly known as nosema. So it's a damp climate. We have big problems with chalk brood and varomorpha. So I need to really keep an eye on that and really document and cull the lineages that are showing up that problem. I like to see evidence of early mating, perhaps, as I said, but I also like to see evidence of supersedure. So I'll go into that in a moment. And I also like to test how long my queens can live. So during swarm control, I take out my breeder queens, I'll put them in a nook box. If that queen is vigorous enough, we'll say in year 2, to head up a colony, so be it, she'll be given a full colony. Or there's any number of things that you can use a nook like that for. You can bolster the new colonies, you can bolster your honey producing colonies. So an old queen in a nook is very valuable in itself, but if you leave that queen until she's not productive anymore, you will soon find out whether you've got supersedure traits in your population or not. And I'd say about 50% of the time, my queens will get superseded. So inevitably, if she doesn't head up a full colony anymore, she'll be given a nook box, she'll overwinter in that nook box, and the following winter, I might be lucky and I might find a new queen. I might find 2 queens actually. I might find the mother and the daughter. So it's nice to breed supersedure into your population, but you won't know whether your bees have got that trait unless you allow your old queens to live on. I like to see behaviors that are sensitive to the immediate external environment. So I don't want bees producing loads and loads of brood if there's going to be a June gap. I need them to be responsive to the environment, and the native black bee is that, very much so. I like to see lots of propolis, even though it can be a pain and sticky and so on and so forth. It's antiseptic, it's there for a reason, and the more of it I see, the better. I always keep an eye on the color of my bees, but in recent years, I'm lucky I can avail of DNA analysis as well. So I would always say to people, consider carefully what you cull. Definitely cull bees that are underperforming, but in terms of color, you could have, it's coming out now in the research that you could have a certain degree of nativeness, but be fooled into thinking that because of the color of the bee that they're not native. So if you can avail of DNA analysis as well, I would really recommend that. So I forgot to mention the pictures here. So we have hazel is one of our earliest pollens. When I overwinter my bees, I don't want to have to feed them fondant. I treat once a year with Apiguard, and I put the eeks on and I leave them on. I take the second tray of Apiguard away when it's empty, And then I allow the bees to fill that space with ivy honey. And that if they fill that space with ivy honey, I know they'll have enough food to see them through until April. A lot of people would say that's a really messy problem then to face into in April. But another advantage besides not having to feed is that at the center of that nest, the bees like to cluster up there. They have full contact with each other. They're not going to starve. And also you'll see the first drone brood just with a very quick inspection. You'll see when you get your first drone brood, and if you want to open up some of the brood, you'll see what your varroa levels are like. So for me, I find it a really good extra management trick, if you like. When I do open up and I see the first drone brood, I will consult my colony notes to see which colonies I wanted to breed from. And then at the very first inspection, inevitably the brood nest is still quite small, but expanding nicely. So you can measure that rate of expansion over a 2-week period, if you like, and compare it with your other colonies. Inevitably, some of them will be, on 7 or 8 frames of brood before others. So they stand out as your breeders, even without consulting the notes. But I will consult the notes. And at that point, then I'll take out some old comb that would be kind of earmarked for rendering. And I'll cut out the bottom 2/3 now. I was doing 1/3, but now I do 2/3. So I'm basically just leaving a strip of comb with honey in the top. And if the bees are ready, I'll position that then at either, depending on the size of the cluster, position it at frame 2 and 8, or maybe 3 and 7. And if the bees are ready, they will draw drone comb and the queen will lay in it, because it's nice, clean, fresh comb. She'll lay it up with drones. So I'm controlling my drone population right there. And in 40 days' time, I know I'll have queen cells, and I know I'll have my own drones. And I've scaled it back a bit now. I have 20 colonies, but maybe 5 or 6 years ago, I would have had maybe 4 or 5 out apiaries with 5 colonies each. And they would have been in a circle about 2 miles away from my home apiary, which is where I do my queen rearing. So all of the drones in a circle, if you like, around my home apiary would have been flying then hopefully to the home apiary for mating. And that does 2 things. So I know I'm controlling my drones, but I'm also almost creating a drone barrier. So other drones are less likely then to dominate, we'll say, in my mating program when I can do that. Luckily now there's 1 or 2, like there aren't many beekeepers in my area. The ones that are in my area, I've generally taught them. And at the very first training sessions, I will tell them about the values of the native dark bee. So, and I'll tell them to buy their bees locally. So that now, even though I have fewer apiaries myself, my neighboring beekeepers I know have dark bees as well. So their bees are now we're feeding into each other's breeding programs now. As I said, queen cells, then I can expect them from the 10th of May onwards. And then hopefully the drones will be in the air the following week. And I can very often have queens laying by the 31st of March. So just to know your bees and know the climate and just document from year to year. And you probably see that the pattern, there's a consistent pattern. One word of warning about rearing lots of drones. If you're rearing lots of drones, as you know, the varroa can reproduce, gets fit in some extra cycles of reproduction in the drone cells. So you're breeding varroa as well. So you've got to embark on a monitoring program. If you, that's just a risk with breeding extra drones. So the top 3 diseases, as I said, that I struggle with here in the west of Ireland is varroa, which is classed as the #1 problem still, varymorpha, which was known as nosema until recently, and chalk brood. So if I chill my bees, I'm providing perfect conditions for chalk brood spores to germinate, and with drones on the outer periphery of the brood nest, they're the ones that are going to show the symptoms first. I'm never going to have nice if I was to measure my success in frames of honey, I'm never going to be a successful beekeeper as long as I tolerate these 3 issues. And that's just a picture on the top left corner of my notes. So a notebook for every year, but then I can cross-reference and track the queens right back to the, at least 3 to 4 years historically. So I'm going to cover some basic genetics that I felt enhanced my beekeeping. So hopefully the same will be true for yourselves. So it's important to understand hereditary and haplodiploidy, which I'll come back to. That encourages a strong focus on your drone breeding and your queen breeding. Obviously, you've got to know your swarm control inside out, pick a method, as they say, and stick to it. And why do I say that? Well, if you lose a swarm, you're going to lose your queen if she's clipped as well. So, and you're going to lose the half the colony if she isn't clipped. So we don't want to lose our breeder queens. We value our breeder queens. As I said, I explained that if you can test for longevity traits by observing how long your queen lives. If you've got a longevous queen, chances are that your workers are also carrying that trait. So it also unmasks the supersedure traits that I like to have in my population. Supersedure is such a convenient way for queen replacement. And queen replacement is inevitable anyway and necessary, but with it comes a genetic reshuffle. So how does the new generation differ from the old until the new generation and the new queens prove themselves? It's also a good idea to hang on to the older queens. I've often had cases where I actually preferred the mother queen to the daughter queen. Understanding mating behavior fosters control of queen mating. So we're going to take a lot more interest in the drones that are flying and what's the best time of year for queen mating. And I think personally that fast tracks success in bee improvement. So for me, knowledge of basic honeybee genetics, it helped me to arrive at my current management regime of continuous improvement. And after several years, the amount of effort you have to put in goes down because your population is improving. It's becoming locally adapted. Hopefully you don't have neighboring beekeepers that are bringing in hybrids. And there's always a tendency when you start beekeeping, particularly in the west of Ireland, that everyone else's bees and queens must be better. They seem to be more prolific, more productive, and so on and so forth. But once you get comfortable with your handling skills improve and your varroa management and your swarm control is good, then you'll definitely, once you embark on bee improvement, you'll see results within 3 to 5 years. And the amount of effort to achieve improvement goes down. Personally, it helped me to stop worrying about inbreeding. I haven't brought any new stocks of bees into my system for 7 years now. I don't see any inbreeding. And as I said, there aren't many beekeepers in my area. So that was one thing less to worry about. And to decide that there's no need, as I said, to bring bees or queens in from elsewhere. The raw material that as long as they're not really prone to certain diseases, what you start with now is probably just as good as stocks that you bring in and start bee improvement on. And in fact, as I said, bringing in new stocks can be damaging to local breeding programs. So just to describe the DNA molecule, I'll go through the structure of DNA. It's not going to make a huge difference to your beekeeping, but everyone's talking about DNA these days. Most lectures, most sort of honey shows or workshop weekends will incorporate a lecture on DNA. And so there's nuclear DNA and mitochondrial DNA, and they're both significant. I'll go to that in a minute. But some terminology first. As I said, DNA is deoxyribonucleic acid. If you have to do a one-liner, I would say it's a complex polymer containing a unique genetic code. And again, I'll do a single slide on DNA in a moment. A chromosome is a structure made up of proteins and DNA, which is organized into genes. In diploid organisms, they're arranged in matching or homologous pairs. And a gene is the basic physical and functional unit of heredity, again, part of the DNA. Genes act as the blueprint for all 20 naturally occurring amino acids. And so groups of amino acids in all possible combinations make up peptides, and groups of peptides make up proteins. So that's what DNA is all about. It's the blueprint for the proteins. An allele is each of 2 or more alternate forms of a gene found at the same place on a chromosome. So generally we're used to talking about 2 alleles, but I'm going to bring up the sex alleles. And the last time I checked the research, I found that there's up to 70 sex alleles now. So that's very important to have a very diverse range of alleles, sex alleles in the population. That's what prevents inbreeding. That's when you know you've got good diversity. But we'll come back to that. And a genotype is an individual's collection of genes. So the term can also refer to the 2 alleles inherited in a particular gene. So back to DNA, the structure of DNA and its function. So it's a blueprint for the traits or characteristics of an organism, and it's unique to every living organism. And it's found in the nucleus of every living cell of higher organisms, and it's constantly being regenerated in living organisms. And as I said, it gives rise to every protein in the body, and proteins make up every living cell, tissue, organ, pheromone, hormone, blood, you name it. Proteins are the building blocks for all of these. So if you were to look at the strands of DNA, what you'd be looking at is a double helix consisting of a polymer composed of 2 polynucleotide chains. So they coil around each other then to form a double helix when the DNA isn't being replicated. And that coiling it helps to condense the molecule. So if you stretched out a chromosome, the DNA is so highly coiled, a chromosome could measure 5 centimeters, but obviously you're talking about nanometers and microns when you're talking about the size of a nucleus. So it's highly coiled and wound and bound by proteins. The proteins maintain that shape. And so it's a very dense material, and its potential is huge then for the amount of information carried on it. So each strand consists of a sugar, 5-ring sugar. There's a phosphate group attached to it, and then we have these bases. And in DNA, there are only 4 kinds of bases, but they complement each other. So there's only two that can bond. So adenine and thymine bond, and guanine and cytosine bond. So you can have any amount of variation in that sequence, and that's what gives rise to different genes and different proteins and diversity. So as I said, that's 1 strand, and those base pairs, the way they bond is through weak hydrogen bonding. So that's what maintains the double helix shape. Nuclear DNA then is found, the nucleus is found at the center of every living animal cell, and it contains the chromosome structures made up of DNA. And as I said, it's compactly stored in the form of highly folded and tightly wound strands. And it's inherited from both parents, and it's the recipe for the physical and behavioral traits of an organism. There is such a thing as mitochondrial DNA. Mitochondria are little organelles in the cell, which they say originated from archaic bacteria that got incorporated into the first animal cells, but they're now responsible for producing the energy in a cell. So it's a much smaller molecule of DNA. It's circular. It's still a double helix. And when we talk about the length of DNA, we talk about how many base pairs. So there's 16,463 base pairs in the mitochondrial DNA of the honeybee. And 1 base pair is that sugar and phosphate and base pair, that molecule, just 2 sugars bonded with the phosphate groups, and and the base pairs holding it together. So that's one base pair. The length in base pairs of a chromosome would be orders of magnitude bigger than that. So just so you know, the mitochondrial DNA doesn't confer any physical traits. It just encodes genes and therefore proteins that are involved in the generation of energy for that cell. So there are also transfer RNAs involved in synthesizing. So it's a self-contained unit of DNA which helps produce energy for the cell and helps replicate itself. The interesting thing about mitochondrial DNA is it's maternally inherited. And this is key for geneticists looking at the biogeography and evolution of Apis mellifera. So sperm-derived paternal mitochondria and their DNA, they do enter the oocyte, but normally they disappear during embryogenesis. So we know for a fact that that DNA is inherited, and geneticists can then map that DNA, sequence it, and find patterns, and then denote those patterns to different subspecies. So it has been really helpful for genetic evolutionary biologists to trace the different subspecies of Apis mellifera and other species of bee. So we'll go on now to discuss the unique mating behavior of Apis mellifera, because it ensures genetic diversity. And genetic diversity is the foundation for fitness. So the queen has a very perilous mating behavior. She has to leave the nest. She generally doesn't want to leave the nest. As a virgin, she's encouraged and nipped at and chased around the hive, and she has to leave the nest to go on this mating flight. Lots of things could happen to her on the mating flight. She could get lost, but because of this behavior that's evolved over millennia, it has ensured colony diversity and fitness. So it just has evolved to be the way that queen bees mate, so because the advantages outweigh the disadvantages. So as you probably know, she has to mate with multiple partners. If she doesn't mate with enough, she won't collect enough sperm to last her lifetime. So there's one mating event, but it can take place several times during the day, usually at a particular time of the day, or it can happen several days in a row when the weather conditions are right and when she's the right age to fly. But it's still classed as one mating event. And so she only ever has one mating event. And hopefully that little vessel that holds the sperm, called the spermatheca, will be filled during that event, because if it isn't, she won't be fertile for more than a year or two. And the colony fitness is compromised then if she doesn't mate with enough different drones. On the other hand, the whole system has evolved to protect the queen in a way, because if she mates with too many, if she had to go on way more mating flights than that, there's a higher risk of losing her. And there's maybe in a shower of rain or being predated by birds or whatever. And there's a higher risk of damage to the queen if she has to mate with more than 15 or 20 drones. So the whole process has evolved to guarantee genetic diversity and fitness of the colony. And then, as you know, the drones mate in congregation areas. The queens generally fly further than the drones, so that is one way of preventing inbreeding. That's kind of built into the system through evolution. There's lots of competition amongst the drones particularly. So you get maybe several hundred or 1,000 drones in a drone congregation area, depending on the population of bees in the area. And they have to be really physically robust. So they have to be strong and healthy to be able to fly and get into that comet of that smaller number of a comet of bees that get the queen in their sight and compete with each other to be able to mate with that queen. So they have to have excellent eyesight. They've got huge eyes, as you know, for that reason. And they have to have excellent chemosensing abilities to pick up the queen's pheromone. And then So if he's lucky, he gets to have a single mating event in his lifetime, and then he dies. And so many of them never get to mate, as you know. Drones are very sensitive to the environment. They're very sensitive to pollutants, to varroa, as I said before. More varroa will breed in a drone cell. They're more prone to chalk brood because they're laid on the periphery of the brood nest where the temperatures are slightly lower. And they're also classed as a huge drain on the colony, so only a few hundred are produced in every colony. So drones are very, very valuable and highly regarded. They're welcome into every colony during the active mating season, and then, as you know, because they need more food and can't forage themselves, they're kicked out at the end of the mating season. So some breeding terminology before we get into genetics of breeding. I think I might have said it before, homozygous is having two identical alleles of a particular gene or genes. Heterozygous means having two different alleles of a particular gene or genes. So the female caste, the queens and the workers, are diploid, so they have two sets of 16 chromosomes. The drones are haploid, they have one set of 16 chromosomes. So drones therefore are hemizygous, they carry only one type of allele because they're haploid. Phenotype then is an individual's observable traits, simply put, things like body size, eye color, and so on. But in beekeeping, phenotype is better described as morphology, development, biochemical, or physiological properties and behavior. So all of those things are governed by genetics. Variability is being expressed as genetic plus environmental variances. So epigenetics is a whole other school of genetics, if you like, and depending on where your bees exist, different traits have to be present. So therefore, different behaviors have to be seen: early foraging, late foraging, early mating, multiple mating, and so on. All of those are behaviors, but all of those behaviors are genetically coded. But some genes are switched on and some genes are switched off based on the immediate environment. So that's epigenetics. Dominant genes code for traits that are predominantly expressed, and they show their effect even if only one copy of the allele is dominant, in other words, heterozygous. And then a recessive gene is a gene whose effects are masked by the dominant gene. Some other two terms that I'm going to give a worked out example for, but I'll describe them here first. F1 is the first filial. So for example, if you if you're starting your beekeeping journey and you bring in two different lineages of bee and you want them to mate, and you can do it under controlled conditions. This would normally mean instrumental insemination and so, or remote mating. Then if you mate a homozygous dominant mates with a homozygous recessive, all of the progeny will be 100% heterozygous. If you then take the progeny from that initial mating and mate them, so in other words, in a lab in a setting where you actually want to breed with, say, a recessive gene, it involves a certain amount of inbreeding. So it is done for the likes of varroa, sensitive hygiene, that kind of stuff is only achieved through very controlled mating, and it usually involves instrumental insemination. So this is a bit theoretical, but the second filial generation cross of heterozygous female with hemizygous drones yields a 1:2:1 ratio. And I'm going to show you a worked out example of that, but it is helpful to remember. So if you embark on a journey whereby you think, oh, I'm going to focus all my energy on breeding for varroa, sensitive hygiene or hygienic behavior, I think it's widely known that they are recessive traits. And you could end up putting an awful lot of energy into doing that. And at the end of the day, all you can achieve is that 25% of the time for a cross like the one I've mentioned, you'll have homozygous recessive. So you're putting a lot of energy and you're not getting much results back. So for me, understanding genetics helped me to just sort of leave that work and concentrate on improving the traits I mentioned to you earlier. So I don't bother trying to breed for hygienic behavior because the results are very slow, and unless you can guarantee or avail of instrumental insemination and do some inbreeding, it's very hard to get those traits embedded in the population. So some worked out examples of that would be, so the first one, example one, I said the first filial is a genetic cross of a homozygous queen. So we'll take, for example, a very visual trait such as eye color, and big B stands for dominant, which is black, and small b stands for brown, which is recessive. So if you take a queen who's homozygous for black eyes and mate her with a drone who has brown eyes, which is a recessive trait, the F1 filial progeny will have, you see that line there? Can you see my pointer, by the way? Sorry.
No, we're not able to see the pointer.
Okay, so under that first, so these are called Punnett squares. So under that first Punnett square, it says F1 females. So all the female progeny of that first cross will be big B, little b, and so that's 100% heterozygous, and the color of eye will be black, but they're all carrying the recessive gene. So, and by the same token, all of the drone progeny will be 50% dominant black eye and 50% recessive for brown eyes. So there's a 50-50 ratio of the trait in the males, and then there's 100% heterozygosity. All the females are carrying the recessive trait. So if you were to take those females, and some of them were to become queens, and you take a queen with that genetics, big B, little b, a carrier for the recessive trait, and mate those queens to their sibling brothers. So again, this does happen. It's how they're researching the hygienic behavior, and it's done with instrumental insemination. So now you have two Punnett squares, because half of, as I said, there's a 50-50 ratio in the F1 drone progeny. Half of them carry, half of them are have black eyes, half have brown. So we have to have two Punnett squares and do a theoretical mating with that heterozygous queen who's carrying the recessive gene. And now at the bottom of the two Punnett squares, you'll see the outcome is big B, big B. Then you have two big B, small b's, and you have one small b, small b. So you have one homozygous dominant queen with black eyes. You've got two black-eyed queens that are carrying the recessive gene, and now you've got one recessive. So you've sort of pulled out of the population through mating, you've pulled out that recessive, and it's expressed in a female. And if that female becomes a queen, then you can propagate that recessive. But it's very hard to maintain recessive genes in a population. This is a very artificial situation, but it is how desirable recessive traits are being bred into populations. So overall, the proportions there would be 25% homozygous dominant in the female cast, 50% heterozygous dominant, and 25% homozygous recessive, which wasn't in the population before. So I know that this is probably the heaviest part of the lecture, and we can come back to it if you want, but I just wanted to show you worked out versions of those genetic crosses. And as I said, that's just one example. So now I'd like to spend a few minutes on discussing how genetic variation is part and parcel of reproduction and colony propagation. So oogenesis is the formation of the egg, and so somatic cells are the cells in an individual that gives rise to the gametes or the sex cells, and in this case the oocyte would be the female gamete, and the sperm is the male gamete. So the egg cells undergo what's called a reductive division. So the eggs start out, the queen starts out as diploid, but when her eggs are formed, they have half of the genetics of the queen, a random half of her genetics. So that's called a reductive division. So the cells are dividing, and the genetics in the final outcome is half of the mother's. And so therefore, in the traditional sense, when a sperm and an egg meet, then that reductive division is restored to its original state, which is diploid. It's slightly different, as we know, in honeybees. So during that process, we'd say there's 4. So one somatic cell gives rise to 4 egg cells, but in reality, in the animal kingdom, 3 of those never become the oocyte, only one of them. That's just a minor point. You don't really have to worry about that. During that process of meiosis, though, are some events, and one of them is called crossing over, and the other one is called recombination. And these are events or processes that introduce an awful lot of diversity into the genes and into the chromosomes. So for that cell division to happen, the first thing that happens in the somatic cell is that all the chromosomes line up in the center of the cell in readiness to multiply and then divide into 4. So I'm going to give you, so I'm not going to, there's a lot of phases in meiosis. I don't want to get bogged down in that. You could spend an hour on that. But just so you know that when the chromosomes, the homologous pairs line up, there must be little electrical charges, because sometimes chunks of one chromosome can literally swap over and join the other chromosome. So straight away now, the daughter chromosomes are different from the mother. And likewise, the chromosomes that were, when the cells divide into 4, those chromosomes don't stay in their original arrangement. They can swap over. So you've got chunks swapping over, and you've got the paired chromosomes swapping place as well. So that guarantees that the daughter cells are always going to be different from the mother cells. And those daughter cells become eggs, and unfertilized eggs become drones. So therefore, that's how diversity is introduced into the drone population. So every drone is unique because it arises from an egg that's unique. And so those drones then become a genetic ambassador for the queen. So on the wing and in flight, you could visualize them as flying gametes. The one thing to remember about is when the drone produces sperms, that's spermatogenesis, and normally in other animals, that's a reductive division too, but the drone is already haploid. So the second phase of meiosis is aborted in spermatogenesis, and so the haploid state is maintained. It's not divided further. Again, that would be the second most sort of trickiest part of genetics to understand. It's so different in hymenopterans and social species. It's called haplodiploidy, but it has evolved to guarantee lots of, so that rate of crossing over and recombination is much higher in the honeybees and other hymenopterans. A little bit of maths now, but we'll make it simple. So as you know, I said there are 16 pairs of chromosomes, and they divide up to form gametes. So there's 2 possible versions then of every combination of 16 items. If you're to do the maths, that comes out at 65,536 possible gametes. And as I said, an unfertilized egg becomes a drone. So that's 65,536 possibilities for a drone. So you can say, just by that maths alone, that every single drone that the queen ever lays is going to be genetically unique. And then, of course, you've got the recombination, which adds to the diversity. But those figures are a bit hard to deal with. So if you think of a theoretical organism with just 3 sets of chromosomes, 3 homologous pairs. And if you just draw out on a piece of paper how many different combinations of the black and the red, A1, 2, and 3 there are, you'll find that there are 8 combinations only. So that works out as 2 to the power of 3. So for 3 chromosomes, each with 2 ways of being arranged, the maths would say that's 2 to the power of 3. So you can take that maths and apply it to 16 chromosomes. And so that's how you arrive at 65,536 possibilities, because 2 to the power of 16 is that figure. So that's genetic diversity covered through mating behavior and recombination and crossing over events. Now we talk a little bit about sex determination, and again, genetic diversity is very important here. So honeybees don't have a gender-specific chromosome like human beings or other animals do. Humans have the X and the Y chromosome. Bees don't have that. The genome instead contains an area called the sex-determining locus. And this region of DNA, which harbors the complementary sex determiner of the CSD gene, or sex allele. And so there's many versions of this allele. Early studies suggested maybe 20 versions, but the last time I checked the research, it's now up to 70 different sex alleles. So it's great to have a population with more sex alleles. And the way that these alleles combine creates the primary signal of sex determination. So going back to the terminology, heterozygosity and hemizygosity. So a heterozygosity generates an active protein that initiates female development. So in other words, if you have 2 different CSD alleles in a female individual, in an individual, it's going to be female because it produces a protein that activates another gene called the feminizer gene. So I don't want to go into all of that detail. It's a bit of a domino effect. But as long as you have 2 different sex alleles, you'll have a female. And so the drones, as you know, are haploid, so they can only be hemizygous, so 1 copy. And 1 copy doesn't produce a protein, an active protein. So default development is male then. So if the individual is homozygous at the CSD, this is another scenario, what's called a diploid male develops. And as you know, diploid drones, they're not tolerated. You have worker policing going on. As soon as the egg hatches, they're cannibalized. So at the first larval instar, if a diploid drone develops, they're eaten. So if you get a lot of that happening in a colony, you're going to get a lot of blank cells. And so that would be your first indicator that you might have a problem with inbreeding. And so in diverse populations, different combinations of sex alleles are more numerous. So the extent of diploid drones is actually an indicator of genetic diversity. So there has to be a protein product from both alleles which combine to activate and commit the larva to the female cast. And there's lots of references on that. I'll go through 2 very simple and extreme examples to show you what I mean. So if a queen mates with a number of drones, we'll say 5. So each color, different color there represents a different sex allele. So on that chromosome where the CSG gene is located, each drone has a different version of that allele. It's not dominant, it's not recessive, it's just a different version of the allele. The queen can only be heterozygous for the CSD gene, and in this case, she has 2 different alleles. They all differ from the allele version that the drones are carrying. So if you do that Punnett square cross that I showed you earlier, you have the S1 gametes and the S2 gametes from the queen, each combined with S3, 4, 5, 6, and 7 from those 5 drones. So the end result are all different, all unique combinations of females. And again, this is a very extreme example, but in this second situation here, it shows you what happens when a queen mates with a drone that shares 1 sex allele with the queen. So it could be a sibling mating. And the result here then her gametes will split into S1s and S2s, and the drone is carrying S2. So when you do your Punnett square and cross all the possibilities, you'll find that 50% of the population is normal female workers and 50% are diploid drones. And like in reality, this never happens, because that would mean 50% of your population is being culled at that first larval instar. But if it's one drone out of 15 that mates with the queen, and that drone is related to the queen, there would be a certain amount of pepper pot brood pattern in a situation like that. So it could be an indicator of inbreeding. So if you want to measure brood viability, that's a way of putting into practice the maths that I've just described. So if you cut a piece of card or plastic out that is equivalent to 100 squares in total, so 10 by 10, and usually we go for a rhombus shape, and you put that down randomly on the brood and mark it out and count how many blank cells there are. And so that's 100 cells, and subtract the blanks from 100. Then you have an approximate value for brood viability. It's better to do it 3 or 4 times and take an average result, and you have to stay within the center of the brood nest. But that's a very practical way of measuring brood viability. And anything over 90% is good. It means you don't have any inbreeding. And in reality, as I said, I haven't brought any stocks of bees into my breeding system for 7 years. So at this stage, you could say everything is related. There must be shared genetics, and I don't have any problem with shop brood pattern. So the next section is just sort of presenting that information in a different way, really. It's just a summary again. So that Punnett square, again, as I said, the 1:2:1 ratio. If you know that you have desirable traits and you've selected your drone colonies and your queen colonies, and if you're a visual thinker like I am, you can nearly visualize what's flying in the air. And so you know that only half of the drones of a heterozygous queen with a desirable trait, only half of the drones are carrying the desirable genetics. So that's the only message really I want you to take away from this, is to try and visualize what gametes are flying and relate it back to your notes and your colonies that you select for breeding. So even though the genotype ratio is 1:2:1, remember we took the example of black versus brown eyes, the phenotypic ratio is 3:1. So 3 out of those 4 progeny have black eyes. 2 are carrying the recessive gene, but the phenotypic ratio is 3:1. So as I said, we talked about dominant and recessive genes. There are good dominant genes and there are good recessive genes. The case in point I talked about is hygienic behavior, which is recessive. There are lethal dominants, but we don't have to worry about them because they will never, lethal dominant genes will never survive. They're not passed on. Lethal recessives, the haploids will die. So if there's a recessive gene being carried in a population, it's exposed in the haploids and they die. And so they're purged from the population. And this is why haplodiploidy is such a useful process, if you like, in social hymenopterans. It guarantees that lethal recessive genes are purged from the system. Now, you can also have non-lethal traits that are damaging and deleterious and carried by 25% of the diploids and expressed in 50% of the haploids. That's where we're coming into things like proneness to disease. It's there all the time, and if we are not ruthless in our selective breeding and culling, we're going to propagate it. So a lethal dominant trait is lethal whether the individual is homozygous or heterozygous. So they're never passed on. We don't have to worry about them. And a lethal recessive trait is carried along to the next generation and is lethal to an individual homozygous for that trait. So as I said, in haploid drones, lethal recessives are always expressed. So these genes get purged from the population. So haploid diploidy increases population fitness. And this is just another representation of that extreme situation I showed you earlier. We'd say this time how a population can harbor recessives, be they good or bad. So in this case, it's an example of a genetic cross which allows a recessive gene to be hidden in the next generation. And the gene, we'd say for black eyes, is dominant. In this case, I said black or red, but I've gone through this in the early Punnett square. So the heterozygous parents, queen crossed with, we'll say, the dominant gene for black eye. So 50% of the progeny are carrying that. So that's something to bear in mind, whether it's a recessive lethal gene or carrying an undesirable recessive or a desirable recessive. So the message there would be, you have to cull a certain amount, otherwise you're just going to propagate, that gene is just going to crop up in the future if you don't deal with undesirable traits. So to summarize a lot of these recent slides, meiosis gives rise to eggs, and recombination and swapping over introduces diversity. Every drone is unique, but his sperm are all identical. So the drones are ambassadors of the queen. Weak drones with deleterious genes won't mate. They're not fit enough to compete in that comet and mate with the queen. So the mating behavior knocks out undesirable traits. If it doesn't knock it out, haplodiploidy will. Drones inherit a random half of the queen's genome, they're haploid, so recessive traits, if damaging, are purged, as I said. So drone selection is important. This is a bit of a stylized diagram, if you like. I wanted to try and map out maybe 2 or 3 years of mating, but in reality, that's impossible to do. It gets really complicated when you try and do diagrams with, we'd say, crossing the drones here with 3 different freshly introduced lineages. So that's a scenario that you might start off when you're starting a queen rearing group. You might bring in different stocks from different areas, hopefully from the local area, but they're all different lineages. As I said before, after 3 or 4 or 5 years of controlled and isolated mating, you could argue that everything is related. But I can tell you from experience, and there are other people who can tell you as well. I mean, we have the example on Colonsay Island and several other isolated mating stations. I don't have a problem with inbreeding, and I've been using the same stocks now for 7 years. And in terms of helping you decide where to put your energies, Alphen and Furno in 2020 have stated that the pan-mating nature, in other words, free-flying and open mating, the pan-mating structure of honeybees prevents local natural selection for resistance. This is resistance to varroa resistance, we'd say. But because resistant genes disperse into neighboring populations at a rate higher or equal to the local rate of recruitment of these genes by selection. So that's why I'm saying that the work on hygienic behavior is generally done under very controlled conditions. And here is an example of a paper from 2019 describing how they are managing to embed that recessive trait into a population. First with 2 rounds of instrumental insemination, and then subsequent open mating, but again in a very isolated mating station. So 3 years of work. So my message here is, where are you going to put your energies into? Everybody believes in continuous improvement and selective breeding, but in reality, personally, I stuck with just breeding for the desirable traits that I described earlier. I no longer put too much energy into trying to breed for hygienic behavior. I have seen uncapping happening. I was a very new beekeeper at the time. I have photographs of uncapped purple-eyed pupae, but in fact that colony died. It was so prone to varroa that it died. So to me, that act was a final act of desperation to try and control the varroa numbers, and it didn't succeed. Again, I would follow very practical beekeepers with a scientific background like Randy Oliver. There's so much information on YouTube. You've got to be careful who you believe and who you follow. And of course, other countries are different from Ireland, so it's always going to be taken with a pinch of salt. But Randy would say with open mating and meiosis dictating how desired traits are distributed in a population of controlled drones, only half the drones from selected colonies will probably carry the desired resistant traits. So I think I showed you that in the Punnett squares. So a breeder can only expect to increase the desired selected genetics by half year on year. So what are the take-home messages? Well, to me, local adaptation is essential, and it has helped my beekeeping enormously and improved my productivity. Apiary vicinity mating, I didn't go into that too much, but in the west of Ireland here, the chances of getting good weather 5 or 6 days in a row for a queen to mate are pretty much nil. So you do have situations where the queen doesn't fly so far, or she may mate in the home apiary where the drones are propagated as well. That's called apiary vicinity mating, and it's better than no mating. So it is an advantage. If the weather is bad and all of the drones are ready to mate and your queens are hatching, it's better than not mating at all. DNA analysis is key. Dark bees can end up being hybridized, and banded bees can actually have a good proportion of native genetics. That's what's coming out of Professor Grace McCormack's work now. So if you can avail of DNA analysis, that would be great. So this is a drone that dropped to its death onto a hive roof, and I deduced from it that it had mated locally. So it does happen. In terms of management, my colonies have to be prepared for all weathers. And as Brother Adam would say, if and when a flow comes along, you have to have colonies ready for that nectar flow. So in June of 2022, it was looking like a terrible year for our bees. And yet 1 month later, our supers were stacked high. So that's how quickly the weather can change. I guess my message here is, so you know your own area, you know what to expect, and have your bees ready. Another thing that I've stopped doing is believing everything I read in the books. So I have some fields around the house that I don't spray. I try and let the wild flowers come, but inevitably buttercups seem to dominate. But in June, when there's a June gap, lots of rain, not much happening, I've actually seen bees forage on buttercups. So again, you just don't believe everything you read in the books. Again, looks aren't everything, and DNA reveals a lot more on the extent of hybridization than just visual. As you know, the founding member of BIBBA would always have said continuous improvement must always be part of the beekeeper's plan. So after 5 or 6 years, even though the effort involved is smaller, you can never give up on continuous improvement. So that's my talk really. I just want to give you a quick update. Thanks to John Greenaway for filling me in. I was more actively involved a few years ago in the Native Irish Honeybee Society, but I haven't been able to be on the committee in recent years. So the website, if you want to read about it, is www.NIHBS.org. And the Galtee Bee Breeders Group began its work of conservation of AMM over 30 years ago, 1991. Micheál McGillicuddy was central to that. And I believe that Beowulf Cooper and Micheál met several times and appreciate each other's work. And 20 years later, this work had evolved into the formation of the Native Irish Honeybee Society in 2012. And NIHBS now has close to 800 members, and it's active throughout the island of Ireland, north and south, and we encourage international links. So when the vision of conserving the various strains of A&M in Ireland was first proposed, a key component was the creation of voluntary conservation areas, VCAs, because we don't have legal protection for AMM in Ireland. We just have to go with voluntary protection. So educating the public and the beekeepers of the country and encouraging them. So that by 2021, there were approximately 20 VCAs. And in each of these VCAs, the beekeepers have to declare against the import of non-native honeybee queens and colonies. And they're engaged in education for the conservation of the native Irish honeybee, particularly with respect to queen rearing. And they're committed to exclusively using native AMMs within their catchment. And then during 2022, a commitment to a threefold campaigning strategy was agreed. So firstly, a political campaign against the import of non-native honeybees. And we're constantly engaging with our politicians on this. And so the protection of the native Irish honeybee bill was read earlier this year, and it's being considered now in the Seanad. Secondly, queen rearing groups have been set up around the island to promote the skills of breeding native queens and education on AMM conservation. And that's been a really successful, tangible effort, a real commendable effort by NIHBS, and it's showing real measurable results. We have one such breeding program in Mayo and one in Galway, and other counties might have one or two. So it's really gathering pace now in the last 3 years. And thirdly, voluntary conservation areas have become conservation areas. And so the variety and quantity of CAs has undergone a rapid rise in numbers because they now include environmental organizations, leisure hotels, schools and colleges, reforesting. So there's a lot of community engagement now. And so that to date, we have 50 conservation areas now, and the numbers are literally growing monthly. So that's just a very quick update from the Native Irish Honeybee Society. And I'll take any questions that you have now. So thank you very much. I really appreciate your attention.
Thanks very much, Helen. We've got 2 talks in one there. Well done.
Well, okay, I'll give you that. Okay, I do. I'm not known for brevity, as you know, but there's a lot to fit in, and I struggle when I have to leave something out. So that's to be honest.
Okay, we've got you've actually only got one question at the moment. So if you've got any questions, please fire them in. The first one is you mentioned using swarm cells for new queens. Many people believe this method simply propagates swarmy genetics. Is this correct?
Yeah, I referred to that briefly. So the native dark bee doesn't produce huge numbers of swarm cells. So it's a little bit of a compromise, if you like, and I've arrived at that decision over several years. I've tried lots of different ways of breeding my queens. And I have found for me, and again, this is a very personal account of my beekeeping, I found that queens from natural cells are the best. And I'm not in a position where I have to supply a lot of nooks to beginners. I'm not relying on that income for an income source. So I'm open to whatever works for people. But for me, I find that the queens that emerge early in the year from swarm cells are my best queens.
Well, it's your talk, not mine, but I think I have to agree with you because, I mean, the bees have been using natural cells for millions of years, really. And I think it's probably to do with the number of queen cells that are produced at the time of swarming. And I've noticed that the more queen cells, the more swarming the colony. The less queen cells, the less swarming.
Yeah.
Anyway, there's nothing else coming so far that I can see. Just a couple of things from me. One is you showed a sort of split screen very early on of some bees with pollen on their legs. A huge number. I've never ever seen anything like that.
Really? That's gorse and dandelion when the two coincide.
I saw the gorse there.
Yeah.
but you certainly haven't got a pollen shortage, have you?
Definitely not. And we have willow as well. So the village I live in is called Graffy, and a loose translation from Irish would be low-lying land of scrub. So it's quite insulting to farmers, but for me it's ideal because I have loads of willow, a lot of gorse, and then the pollen, and then the dandelion comes in.
So you actually mentioned your area. What do you reckon it's like for honey? I mean, is it marginal, or is it
Well,
middling,
yeah,
or
we get a modest crop of honey, I'll be honest with you. So this past summer was our best since I started beekeeping, and I got 4 supers. So to a lot of people, that would be very mediocre. I mean, I've been examined by people from BIBBA, from other parts of the country, who said if that's the crop they were to get in a good year, they wouldn't bother keeping bees. So it's all about your expectations. It's about what, you know, and then if you're into honey gathering, there are ways of artificially bolstering honey producing colonies. I tend to leave things as natural as possible. But I do know of one commercial beekeeper in Connemara. So it is possible to be a commercial beekeeper and get a good crop of honey. And as I said, it's down to management. And what Brother Adam said, you know, you have to have your colonies. It might take a bit more management. You've got to have your colonies in a state whereby they can grab that nectar when it comes, you know. But you need to be skilled in this part of the world to get that crop.
Right, there are a couple coming in now. You mentioned the use of mitochondrial DNA to differentiate different subspecies. Could you expand?
I'm not going to expand. I'm not really going to bluff that much. My area is not pure genetics, but it is a fact that because the paternal DNA isn't incorporated into the developing embryo, that you can trace lineages through the female line. So an expert in this area would be Grace. I won't expand beyond that point, but it's just the way that the biogeographics has been established. It's through the maternal mitochondrial DNA.
The next one, is it possible genetically to prove the existence of a native Irish bee? Lots of discussion on Facebook recently that has totally confused me. What proves a bee as native?
Well, again, I mean, the expert there would be Grace and her team. It's down to reference DNA is how they measure. So they're taking samples for the paper that was published by Jack Hassett and Keith Brown and co-authored by several more, Grace included. They took samples of DNA from bees all over Ireland, and they measured it against known references. So then they were able to measure C lineage against M lineage. And so they published a peer-reviewed paper that proved beyond doubt that the amount of M lineage DNA in those samples proved that we have a unique haplotype in Ireland. There are always going to be people who contradict that, but when it's peer-reviewed, published work, and I mean, I can supply the reference if you want, but it proved beyond doubt that we do have a unique Irish haplotype of the native bee here in Ireland.
I don't know the reason for the question, but I suspect what's behind it is there's a view that because bees were imported from Holland 100 years ago, that really what's in Ireland is just their descendants.
That's one of the most common counter arguments, but again, I would have to refer you to Grace for exact details on that, but they were able to prove that we have a unique haplotype in Ireland that didn't originate from the Dutch bee, which was brought in when Isle of Wight struck.
Well, if you go back before the Ice Age, things all around about the time, bees were moving around all the time anyway. So right, now it appears that Richard's had 2 questions sent to him. So do you want to open up, Richard?
Yeah, so a couple of questions have come to me. First one, is, oh, I've got quite a few that have come through. Sorry. How many colonies does Helen select for drone rearing around her mating apiary?
I would select at least one colony in every apiary. So at the moment, I just have 4 apiaries. So I would always pinpoint one colony. But in reality, if the traits are good, if it's as your bee improvement, gives you results year on year, you're going to have more to choose from. So if every colony in my apiary has good traits and is not prone to the big 3 diseases, I will encourage drones in all of those colonies. So I'll make my final decision in the spring. I'll look at the notes. I'll have a fair idea. So if it meant that every single colony gets 2 frames that's cut for drone brood, that's even better again.
And you select totally unrelated queens for drone rearing?
Well, at this stage, as I said, you could argue that everything is related. I can't tell you now at this stage, because I haven't brought any stocks in 7 years. You could argue that everything is somehow related. But the genetic diversity that's ensured because of all of those events I talked about during eugenesis and so forth, I don't have a problem now. So I don't track that. I don't track that anymore. I'll be careful enough about it, but I don't have to. I know I don't have to worry about inbreeding. I can trace all the lineages, but how do I know what, in reality, I can't tell you what is and what isn't mating with each other, you know?
Helen, do you know what the level of free living colonies is in your area?
I don't think there are many. The habitat, I'd know about them if they were in the area, because inevitably, if it's in a house, someone's going to ring you up and say, there's a swarm, can you get rid of it? There's not much of that happening, and we don't have trees big enough with a wide enough kind of diameter to home feral colonies. So I don't see many feral colonies at all.
You mentioned that hygienic behaviour is recessive. Are there specific characteristics that are dominant and so more easily selectable?
Not that I know of, and the research is very, very much in a work in progress. There's a lot of research on it. Sue Colby would be one, and that other paper that I put up earlier. So it's a cluster of genes, and I haven't heard that any of them are dominant. So for me, that's why I just leave that work to the scientists or people who can. There are non-treaters in Ireland who appear to be very successful, and they take a big hit every year on colony loss. So that would be like a live and let die approach. So no, I don't know of any dominant genes. As I said, we've an awful lot to learn yet in that area. So I would never say there aren't any dominant genes. We just don't know yet.
And then I've just got a couple about DNA testing. So the first one is, where do you get your DNA analysis done? And then the second one is, if you're unable to access DNA analysis, how accurate do you believe wing morphology is?
I suppose I came to the second answer first. I came to beekeeping at a time when DNA analysis was overtaking wing morphology. So I know about wing morphology. I haven't done it myself. I'm very fortunate in that Grace opened up the DNA testing to all students of the Diploma in Apiculture this year, and then she extended it later in the summer to 2 colonies for every beekeeper that wanted to submit samples this year. So that work is ongoing. So I know that I can send samples to Grace anytime and they'll be done. But I think there is, if you Google it, there are some foreign labs that do this as well for a fee, maybe 50 to 100 euros or £100 per colony.
Thank you. That's the questions I've got.
Yeah, I'm just trying to.
There's 3 in the chat. Did you get those, Richard? Yeah. Would you focus more effort on drone selection or improving queens? No, I give the same attention to each. Does Helen select totally unrelated? Oh yeah. At this stage,
Yeah.
So at this stage, 7 years in, without bringing in colonies, I can't guarantee that my bees aren't related, but as I said, I don't get any spotty brood pattern. I don't have any inbreeding. I think the diversity, and that's one message I really want to get across today, is I outlined all the different ways that genetic diversity happens. And so that's reassuring for me. And then not seeing spotty brood, I'm fairly confident I don't have any inbreeding. And then reading the story from Colonsay and so on and so forth, 20 years on an island breeding from the same stocks. So that when I heard about that story, that again filled me with enthusiasm and confidence as well. Right. Thanks to Keith. Oh yes, I see. I think I follow Keith on Facebook. Thank you very much, Keith.
Yeah. Got somebody here. I don't understand why drones show 50-50, big B, little b, when the mother is big B, big B. First
okay, no, yeah. So if the mother is big B, then her gametes are all big B. If you want, I can go back to that.
I think it would help the questioner, yeah, if you don't mind. Whilst you're doing that, somebody's asked a question where they can get DNA analysis done. Look up BBYTES, BEEBYTES. They're from Edinburgh University, are they? Roslin Institute, sorry. And don't quote me, but I think they're about £50 a sample.
Helen, if you want to get your pointer on, if you go down to the bottom corner where the DIA logo is, there's a little pencil. If you click on it, it's just under the A, a little pencil, just very faint.
Oh, yeah.
And if you click on there, there should be an option to do laser.
Yeah, okay.
Yeah.
So this was the first example. I'm not sure if this is the one the person was referring to. So
yeah, first cross, I think.
So the F1 was the example I chose, was 2 new lineages brought in, one homozygous dominant for black eye mated with drones carrying the recessive. So the queen's gametes split to big B and big B, and they're mated with a recessive each from the drone small b. So the result there is every single example of the progeny, they're all 100% heterozygous. And then if you take if one of those females develops into a queen, and you take that queen and you mate it with sibling drones, the drones from that F1 progeny are 50% are expressing the big B, the black eye, and 50% of them are expressing the recessive. So that's why we have to have 2 Punnett squares to describe the potential theoretical matings of the siblings. So again, we have the heterozygous big B, little b crossed with the dominant version of the male gametes, and then we have the heterozygous queen mating with the other 50% of representative genes from the male, the recessive. So when we do all the theoretical crosses there, we end up with big B, homozygous dominant for black eye. Big B, little b happens twice overall. So that's heterozygous, but the eyes will still be black, but they're carrying that gene. And then finally, we've sort of extracted a recessive from the population, small b, small b. So overall, the ratio of genetics is 25% homozygous dominant, 50% heterozygous dominant, and 25% homozygous recessive, which we didn't have in the population before. And that's 1:2:1. But as I said in a later slide, the phenotypic ratio is 3:1, because 3 times there's 3 out of the 4 elements have black eyes. Some of them are carriers, but overall the phenotype is 3:1. I'm not sure if that answered the question though.
Well, we'll soon know, because when it's recorded, the questioner can have a look. So
I'll
right.
stop sharing. So
Okay, yeah. There was one here. Did you answer the one about chalk brood?
I didn't see that question. No.
In your experience, is it possible to select for resistance to chalk brood in a climate like yours? Presumably meaning that it's damp out there.
The short answer is I'm still struggling with chalk brood. So if anyone has a better answer than that and has succeeded, I'd love to know, because I haven't, and I've paid extreme attention to hygiene, to no cross contamination. I've done everything. I've spoken to loads of people in Ireland who pay the same attention to chalk brood. It's really frustrating. Yes, you'll see the hygienic behaviour. You'll see the mummies being thrown out, but really we don't want them to have to be doing that in the first place. So no, I haven't achieved chalk brood resistance at all.
Okay, well, thanks very much, Helen. You've been going for an hour and a half. Well done. And
my apologies if I put half of the audience to sleep. I do apologize.
Yeah, I think I can now reveal that about this time last night there were several emails floating about, and Helen actually shortened it from what it was. So so anyway, thanks very much indeed, Helen, and I'm sure that once people can see the recordings, they'll be able to understand it a lot more. Thanks very much indeed.